Hi everyone! In continuation with the concept about diffusion and osmosis, there is another concept called tonicity. With tonicity, there can be three different situations: isotonic, hypertonic, and hypotonic. This iso versus hyper versus hypo all have to do with the amount of solutes, whether inside the cell or outside of the cell. Iso means equal amount of solutes. Equal amount of solutes on either side, whether inside the cell or outside of the cell. So this is a similar situation as the separation by the container, but now we have a situation where we are talking about a big container with fluid in there, some type of solution, and inside we're going to put a big red blood cell in there. Now red blood cell is known to have 0.9% sodium chloride or salt. So if we're talking about equal amount of solute or isotonic solution, if we're putting red blood cells in a solution that is isotonic, it means the solution also have the same amount of solutes. In this case, 0.9% salt. Because they're same amount of solutes, whether inside the cell or outside of the cell, there is still water movement, but water is moving in as much as water is moving out. Okay, so you will have no net water movement. No net water movement. Okay, no net water movement in this case. When you have a hypertonic situation, it means you have higher amount of solutes. Hyper always means higher. And so in the same situation where we have the red blood cell dunked in some solution with water and salt in there, and red blood cells will not change in the amount of salt they have. They will have that same 0.9% that we saw earlier. Now it's being dunked in solution that contains, let's say, 2% NaCl, okay, sodium chloride. So now there is more salt on the outside of the cell compared to the inside. Remember, what is the rule for osmosis? And that is another thing important for tonicity. We are always talking about osmosis, which means, of course, we know salt cannot just freely enter any cell, okay? Because especially for animal cell, we have phospholipid that are protecting the cell from just allowing things to go inside. So what happens is then it's just water that's doing the movement, which is then osmosis. And so if you have a higher amount of salt on the outside, the rule of osmosis, remember, is water always follow salt. So water will then move outwards into the surrounding because of the fact that there is higher concentration of solutes, in this case salt, compared with inside the red blood cell. Now, if water comes out of the red blood cell, this red blood cell ends up shriveling up, kind of like that. And we call that, we have to give it that process, that shriveling, a specific name in biology, called crenation. Okay, crenation for the red blood cell shriveling up because of the loss of water. Hypotonic solution then means lower amount of solutes. Anything that starts with hypo always means lower. So we have similar situation. Again, some body of water. And now this water is 0.3% sodium chloride, let's say. And we have the same red blood cell that have now still the same 0.9% salt. Remember again, water always follow the side with higher salt. Which side has higher salt? Inside the cell. And hence, water will move from surrounding into the cell. And depending on the concentration difference, the red blood cell will continue to swell, get bigger and bigger and bigger until it just completely gets bigger and bigger and bigger until it completely, in a sense, disintegrates. Completely explodes, for lack of a better word. And of course, there's a fancy term, biology term, for that red blood cell exploding like a water balloon. And we call that hemolysis. Hemolysis for the cell breaking apart. So this is the 3 situation of tonicity in regards to the surrounding solution with the red blood cell. Remember, you have to make sure the problem defines whether the solution surrounding the cell is isotonic, hypertonic, or hypotonic. And that's how you know which way water will move for tonicity.